All right. Welcome, everyone. So today we're talking about mediation. What does mediation mean? It means how are genetic variants influencing their impact on disease. So, namely, what are the intermediate phenotypes that are affected by these genetic variants, and how are those intermediate phenotypes ultimately leading to complex trait disorders? So we're going to be talking about expression, quantitative trait loci. Expression, because a lot of what we're going to see is gene expression intermediates, and then QTLs, because quantitative traits basically means traits that are not simply binary, but just like human height, they are scalar. And loci basically means genetic loci, namely SNPs and haplotype blocks. Okay, so eQTLs are quantitative traits that pertain to gene expression. So eQTLs are genetic variants that affect gene expression levels. Everybody with me so far? Great. So we're going to be talking about mediation and eQTLs. So specifically, on Tuesday last week, we talked about population genetics, about basic variation. On Thursday, we talked about disease association, bringing in phenotype. And today, we're talking about specifically quantitative traits. And on Thursday, we're going to talk about heritability, namely systems level genetics. Okay? So just quick recap of what we talked about in lecture 14 on Thursday. We talked about genetic variation, how we detect it, how do we quantify it, and what are the initial insights, about SNPs, and about haplotype blocks, about how genetic variants are co-inherited and how we can use this to our advantage to map regions. We also talked about how these co-inheritance also had a flip side, a downside, which is that once we figure out the region of association, we don't know the specific nucleotide. And then we talked about how to carry out genome-wide association studies and how to interpret them, how to basically infer the mechanism underlying genetic loci. And, you know, but today we're basically going to be looking at how do we assign function to that. And one of the main things that we talked about on Thursday that I'd like to remind you is how we can, just one second. How we can distinguish different types of variation in the human population. And in particular, the very dramatic difference between these rare alleles that have strong effect and these common alleles that have weak effects, okay? Because a lot of the lecture today sort of relies on this distinction, and specifically that these rare alleles of strong effect very often perturb the proteins directly. So if you perturb the protein directly, you're going to have 100% of that protein's function disrupted in every tissue and cell type where that protein is expressed. Yes, you might have another copy of that protein. Yes, you might have some distant homolog of that copy of that protein that is also, you know, able to perhaps remedy some of that function. But protein coding variants typically have complete disruption if they are a truncating variant, or sometimes a dominant negative effect if they sort of have the ability to bind one partner of the protein, but not the ability to bind another partner. You're suddenly going to be sequestering the function of that protein completely, even if you only have one bad copy of that gene, and so on and so forth. So these rare alleles are very frequently protein coding, whereas these common alleles of weak effect are usually non-coding. Okay, so there's a dramatic difference in frequency and effect size between protein coding and non-coding mutations. Raise your hands if you're with me so far. Awesome, great. So this is the fraction of Mendelian traits that map to protein coding loci. And you can see here that 90% of them are in fact protein coding. These are strong effect, rare variants. By contrast, if you look at complex disorders, like the things that everyone gets, the things that every family gets, then 90% of those are in fact non-coding associations, and only 10% of those in fact hit protein coding genes. Everybody with me here so far? So basically, there's this dramatic contrast between the Mendelian traits, where you have a strong effect variant that affects a protein, and the complex traits that affect the vast majority of the population with huge polygenicity, many different locations, all contributing together in largely additive ways to the risk and largely affecting the non-coding gene. Okay? So one of the things that we talked about is how LD hides the driver variants because of linkage disequilibrium, because of these co-inheritance of large haplotype blocks, and all of the alleles, all of the SNPs in that block having the same allele identity. You basically end up with a nearly identical association between all of the different variants in that region and your trait, making it very difficult to find out which is the causal one. And why is knowing the causal one important? Because depending on which variant is in fact the causal one, you might have a completely different mechanistic hypothesis. Maybe this variant here is acting in, I don't know, immune cell types, and that variant here is active, or that region here is active in brain tissues. Okay, so knowing whether it's this variant or that variant might have very important consequences on the tissue of action, the cell type of action. Everybody with me on this one? Yeah? Raise your hands if you're with me. Awesome, great. Alternatively, one variant might be disrupting a motif for one regulator, and another variant might be disrupting a motif for another regulator. So knowing who the upstream regulator is depends absolutely on knowing what the causal variant is. And then thirdly, one variant might be linked to one target gene, and another variant might be linked to another target gene. Again, finding the causal variant is extremely important. So we don't know the causal variant most of the time because of linkage disequilibrium, because of this co-inheritance of many different genetic variants across individuals. So what we can do instead is look at epigenomic information. We can basically overcome this linkage disequilibrium problem, this haplotype block problem, by basically saying, within the region of association, which of these genetic variants have functional annotations that are perhaps enriched across the entire genome-wide association study. So if we have hundreds of loci, and in 50 of those, you basically have overlap with an epigenomic annotation that indicates activity in immune cells, then you might say, okay, great, I'm going to prioritize immune annotations for any one individual locus. This is one example where I'm learning from the whole genome that this particular trait typically overlaps this particular type of annotation, and then I'm turning around and applying it to any one individual locus by basically saying, now that I know that genome-wide immune functions are enriched, if within the region I find some SNPs overlapping brain and other SNPs overlapping immune, maybe I'll focus on the ones that overlap immune with higher prior. Okay, so the posterior probability of action of any of these genetic variants being causal can be estimated from the genetic evidence, which is given to you by GWAS, and by the epigenomic prior, which is given to you by an enrichment across the genome of all of the genetic variants associated with that trait. Raise your hands if you're with me on this whole sort of prior posterior evidence setup. So the same way that we've done Bayesian analysis throughout the semester, we can now again bring in a prior that is an empirical prior, which is estimated directly by the enrichment of the observed genetic variants on the different classes of epigenomic annotations. Everybody with me so far? Great. And that can basically give us the posterior probability of each genetic variant being causal, which can basically boost up genetic variants that would otherwise be identical and bring down the posterior probability of genetic variants that are not sitting in these enriched annotations. Sounds good? So again, the challenge is humongous. Basically, we have more than 100,000 associations. When I last checked, there were 120,000 associations with complex traits, and we need to sort of understand them. And basically what we need to do is figure out what is the target gene, what is the causal variant, what is the cell type of action, what are the relevant pathways, what are the mechanisms, okay? So this is basically the review from how we do that from the last lecture. So that at every locus, we can now start painting more complex images of who are the upstream regulators, what are the downstream target genes in green, what are the potential causal variants among the ones shown here in red, and what are the tissues of action, okay? And that's where, so basically, as I mentioned, we can basically look for all of the genetic loci associated with any one trait, find all of the enhancer annotations across hundreds of different tissues, and then start predicting for every trait what are the relevant tissues, and use that as your empirical prior for prioritizing tissues of action, okay? So the ultimate goal that we have is to paint a picture like this that basically tells us for every genetic locus, what is the tissue where it acts, what are the target genes that it affects, what is the causal nucleotide, who are the upstream regulators that bind this causal nucleotide, and what are the downstream cellular and organismal consequences. And I mentioned briefly this paper, which I like to joke about, is a paper about 1 bit of information in the human genome about this single nucleotide variant, this T2C variant that disrupts AT-rich motif, resulting in loss of binding of ARID5B, which is normally a repressor. It normally binds the super enhancer, represses IRX3 and RX5, these two downstream genes, which turn out to be master regulators of obesity, master regulators of a process known as thermogenesis or heat generation, which basically depolarizes the mitochondrial membrane, losing energy as heat rather than storing it as lipids, and then switching from lipid storage to fat burning, okay? So this was basically elucidated by using all of these techniques that I mentioned and then putting them together to show the circuitry. And the advantage of finding the circuitry is that you can now start intervening. You can basically increase or decrease the upstream regulator. You can increase or decrease the downstream target gene. You can use CRISPR-Cas9 genome editing to change a single nucleotide. And in each case, you find dramatic effects that basically show that you can restore the process of thermogenesis in risk individuals. You can basically take risk individuals who are unable to thermogenize, who are unable to sort of burn these calories, and by changing a single nucleotide in primary adipocytes from risk individuals, you can basically completely restore the process of thermogenesis. So, I mean, that's precision medicine right there, right? Out of 3 billion nucleotides, you basically say, I'm going to change this one nucleotide. And this one nucleotide has this dramatic effect on the cellular phenotype of completely restoring the cellular function of these adipocytes who are now able to burn energy again. And then altering the downstream target genes has dramatic organismal consequences. So if you go into mice and you decrease these IRX3 and RX5 that are increased in disease, if you decrease them, you basically see that the mice lose 50% of their body weight and they become unable to gain weight in a high-fat diet, okay? So these mice start out leaner, and then when you introduce a high-fat diet here with the arrow, the normal mice gain a lot of weight. These IRX3 dominant negative mice are unable to gain weight, okay? So this is basically the foundations on which we're basically starting today's lecture. So basically what we would like to do is paint this picture systematically for every genetic variant in the genome. So basically what we would like to do is map regulatory variants systematically, use that to develop this mediation framework of who are the intermediate phenotypes that sort of mediate the effect of these genetic variants, and how are these giving us insights into biology. Okay, so that's the goal. The goal is to bridge the gap between genetic variation and disease. This gap is very, very long, and each of these genetic variants has a tiny little effect for complex traits. There are thousands of these variants that together contribute to disease, but the path is unknown. So what we saw on Thursday is this path of GWAS, of genome-wide association studies, is connecting these directly. But what we would like to do as, informants of medical treatment, we would like to figure out what tissue things are acting in, what intermediate gene regulatory changes are happening, what are the genes whose expression changes, what are the intermediate phenotypes, like thermogenesis, that are altered, and how is this enabling us to actually paint a picture, a causal path from genetic variation to disease. Who's with me so far? So the challenge, of course, is that any arrow coming out of genetics, especially for common variants, is usually unidirectional, because my behavior or my socioeconomic status or my smoking or my environmental exposures are not going to affect my common inherited genetic variants. And therefore, I can draw a unidirectional arrow coming out of genetics. However, the environment might actually be affecting several things in this intermediate level. So for example, the environment might be affecting my heart rate, and in response to increased heart rate, my gene expression might actually alter. Okay, my body might be fighting this intermediate phenotype, which is purely environmentally driven, and therefore these arrows might actually be, reverse causality rather than causality. And similarly, the disease itself might be affecting gene expression or enhanced activity or any of these intermediate phenotypes. So we're going to have to figure out ways to cope with this correlation not always being causation. Does that make sense? So basically, it's like seeing firemen at a fire. You can basically say, okay, the firemen are clearly putting on the fires because every time I see a fire, there's a fireman there. You could even, but in fact, it's the firemen who are fighting against the disease. So just because you see immune cells proliferating at the site of injury doesn't mean that they're responsible for the injury. And when you see immune cells proliferating in Alzheimer's, maybe they're not the ones causing the Alzheimer's disease. Maybe they're the ones fighting Alzheimer's. Okay, so correlation does not imply causation. It might actually be the ones who are helping you. So the response is not, okay, great, let's get rid of the firefighters. No, let's get rid of the microglia. No, you have to sort of rewire the circuitry in a way that the behavior of these cells that go up or these genes that go up, in fact, fights disease. And maybe you need to boost them rather than repress them. Everybody clear with these subtleties here? Basically, everybody assumes out there very, very frequently that correlation implies causation. It is not always the case. It's very commonly the case, but very, very often it's not the case as well. So this is part of the reason for mapping eQTLs, mapping these intermediate gene expression phenotypes that are downstream of genetic changes. Okay, this I already mentioned last time. Therefore, we proposed that regulatory mutations may account for the major differences between humans and chimps, but they are also the primary driver from all of the recent research of biological differences between humans, period. Very, very few amino acid differences, but a huge number of functional changes. Okay, so most genetic variations non-coding, we have to go after it somehow. And eQTL mapping basically says, how is this particular genetic variant affecting not the protein directly, but the expression of that protein in particular scenarios, which can then ultimately lead to disease. Okay, so that's what eQTL analysis are effectively doing, basically painting this path of causality. And in the case of this FTO story that I mentioned earlier, eQTLs are actually at the core of this story when we basically found that out of the 6 different genes that sit on one side of this locus and the 2 genes that sit on the other side of this locus, which is folded together in 3 dimensions based on high C evidence, exactly 2 genes change in expression in response to the genotype difference. And again, I can say in response to the genotype difference because the genetic changes always precede phenotypic changes. Okay? So these genes are target genes or E genes of this SNP that is associated with obesity. Okay, so one potential mechanism for that is that the SNP is in fact changing the regulation surrounding these genes and then changing their expression, which ultimately has an impact on disease. The causality might be the other way around as well. Maybe the SNP is affecting disease in some other way, which then in turn comes back and changes the expression of these 2 genes. Okay, so both models should have equal probability in your head, in your mind. You should basically say, well, you know, it could also be reverse causation, could be the other way around, could just be correlation. But what's really helping us with eQTL analysis is that this expression is local. Basically, any kind of reverse causation effect from the disease would be equally likely to affect all genes across the genome. But in this particular case, these genes are next to, or at least near that genetic variant. So out of, I don't know, 3,000 megabase regions in the human genome, you know, they would have 1 chance out of 3,000 of both colocalizing here. And in fact, they both colocalize in that same megabase segment. Everybody with me? So these effects, the fact that they're local, makes it much more likely that the path is through this. But of course, that only implies causality. To verify causality, you have to do interventions. And that's what we did in this particular case. Yeah? So the p-values, how the p-values adjusted? So this is not a p-value, if that's what you're asking. So this is just the effect size. So
no, I mean, there are p-values in the landscape.
I'm sorry. Oh, this or LD? Oh, I see. So we're only testing those individual genes. We're not testing the whole genome. So basically, in this particular case, we just asked how significant is this expression change that we are directly measuring. So we don't have to correct for 6 million hypotheses. Does that make sense? So basically, these are the nominal p-values, and the corrected p-values are basically the same. Whatever you see here divided by 8, maybe, if you want to correct for the 8 genes that we're testing. It's not divided by 10 to the minus 6 or 10 to the 6 of the number of independent LD blocks. Does that answer your question? Great. So basically, the effect size in this particular case is humongous. So with, moreover, these p-values were obtained with, I believe, 3 different replicates, or anyway, a very small number of replicates. So basically, it's not something that you've obtained across, 10,000 individuals or 100,000 individuals, as in the case of GWAS. So, and what you can see here directly is what is the effect size. And the effect size is nearly 2-fold. Okay? Does that answer your question? So basically, for GWAS, the effect size is usually 0.01 or something like that. Whereas here, the effect size is 2. It's like 200 times bigger. Any other questions? Okay, so basically that's sort of what we're after. Okay, we're basically after this eQTL ability. And as I mentioned earlier, we can now actually do perturbations. Making this prediction of causality allows us to go in and perturb these loci, change that SNP, change the expression of those genes, and then infer causality through intervention, okay? So we can basically carry out all of these different modifications and interventions and then ask what actually happens. And if you look at the expression of IRX3 and IRX5, you can basically ask, what is the expression when individuals have the protective allele? And if I use CRISPR-Cas9 genome editing to change this T into a C, what is the expression? And what you can see is a twofold change in expression using that intervention. Everybody with me so far? So that basically says causality. We're done. Basically, we changed that SNP and we saw the expression of those genes change in an experimental assay. So basically, when we take primary cells out of these individuals, we can find that this correlation that we found between genetic variation and gene expression was in fact reflective of causation. Everybody with me? Moreover, you'd like to know that you didn't just mess up something in that cell that led to this expression change. So what we can basically do is a second CRISPR-Cas9 genome editing experiment to change that single C back into a T. And then you see the expression completely restored to the pre-intervention levels. So if you assume that there's some kind of off-target effect of, I don't know, maybe one gene out of 1,000 is randomly hit, when I do this change, I have one chance out of 100 of hitting something else, because this is a different guide RNA, this is a different experiment, this is a different editing. But the chance that this second editing completely reverses some off-target effect that then leads to expression change in exactly that gene is almost minimal. Okay, everybody with me? So moreover, we can basically ask, what is the effect of the gene alteration onto the cellular phenotype? Basically, for each of those, we're testing this model of causality of the SNP altering the gene, altering the cellular phenotypes, altering the organismal phenotypes. And we can ask about this part here of the SNP to gene arrow. We can ask about the gene to cellular phenotype arrow. And doing that, we basically see that if we overexpress IRX3 or IRX5, we basically see this dramatic change in thermogenesis. Okay, so in this particular case, overexpressing IRX3 reduces thermogenesis, just like our model would suggest, whereas knocking down IRX3 increases thermogenesis. Remember this arrow that basically says that this is a negative regulator of thermogenesis. Okay, therefore, increase of IRX3 should block thermogenesis, and decrease of IRX3 should increase thermogenesis. And that's exactly what we find here. So basically, we can test the causality of the gene on the cellular phenotype by direct intervention. And this is true for IRX3, and this is also true for IRX5. Everybody with me? And as a control, we're basically looking at you know, all kinds of other situations where this is actually not happening. So you can see that the cellular phenotypes directly depend on the gene phenotypes. You can also ask about the effect of the SNP on the cellular phenotype directly, regardless of what mediates that effect. And indeed, you find that changing that SNP alone is having a dramatic effect on the cellular phenotype. And then you can ask about the effect of the gene on the organismal phenotype. And that's what I showed you earlier, of basically knocking down the gene dramatically impacts the organismal phenotype. So every time you guys basically do these types of analysis, you're gonna have to think, how do I validate it, okay? And this is one example where we have a whole set of arrows that we're drawing, and we can dissect these arrows one by doing the right experiment, okay? So you should be very skeptical when somebody says causality, unless they basically demonstrate all of these different situations. And the beauty of biology, the beauty of molecular biology, is that you can actually do these experiments. If somebody tells you that, I don't know, eating more bacon causes cancer, you're like, okay, great, there's a correlation, but is there causation? The only way to know that is to go in and intervene in a population and basically say, okay, you guys eat a lot more bacon and you guys eat a lot less bacon, by randomly selecting individuals to be in the eat more bacon group. You can't just simply say, take the class and say, okay, who wants to eat more bacon? And then half the class says, yes, great, you guys eat more bacon. Who doesn't? You guys eat less bacon. The reason for that is that maybe your natural taste buds are adapted to your intermediate phenotype health risk of eating more bacon. And the people who avoid naturally bacon are the ones who are more susceptible to, I don't know, have a stomach ache when they're eating bacon. They may not even realize it. It just might taste bad to them because they had such an adverse reaction when they were kids. So you can't just simply say who wants to do one group or the other group. You have to randomly assign people, no matter how pissed they might be, of being in the more bacon or even in the less bacon group. Sounds good? So that's sort of the intervention that is very often lacking in epidemiological studies. So every time somebody says, hey, coffee causes cancer or coffee causes, I don't know, longevity, you should be skeptical, was it an interventional study? Sounds good? So in molecular biology, we have the luxury of being able to intervene. So that's the motivation for mapping these regulatory variants and mapping these intermediate effects. Let's start going through some examples of QTLs, these intermediate molecular phenotype QTLs, and specifically methylation QTLs in the context of disease. And then we're going to sort of step back and say, what is the conceptual framework within which we can understand eQTL action? What are some insights about, eQTL biology using all of the discoveries? And then how do we complement eQTL analysis with different type of analysis known as allele-specific analysis? And lastly, how do we put it all together in this mediation framework? So this is one example of methylation as an intermediate phenotype in the context of Alzheimer's disease. So across 750 individuals, we basically have genetic variation, we have phenotypic variation, and we also have epigenomic variation on these intermediate molecular phenotypes. So what we basically have is a matrix of 750 individuals, and for each of these individuals, the measurement in post-mortem brain samples of 485,000 locations, and specifically the methylation level for those locations. Everybody with me on what the data matrix is? So this is a longitudinal study. This is not a case control study. These patients were enrolled, and eventually, as they aged, they donated their brain to science after they passed. And these were profiled using all kinds of additional techniques. So the goal is, what are the predictive features of disease between genetic variation, these intermediate molecular phenotypes, and so on and so forth. We also, for each of these regions of the brain, have profiled these histone modification marks. H3K36 trimethylation, H3K9 trimethylation, H3K4 trimethylation, H3K9 acetylation, H3K4 monomethylation. And these are associated with different chromatin states. So we can define promoter states, promoter flanking states, enhancer states, transcribed states, repetitive states, chromatic states, and quiescent states using combinations of those marks. Everybody with me? So here's what the data actually looks like. If you look at a random region of the genome, you basically see all of the chromatin state annotations across different tissues and brain-specific epigenomic annotations of where are all of these chromatin states. Raise your hands if you're with me on the top part of the figure. Great. Then in the middle, we basically have this expression matrix that basically has 750 individuals across 400,000 SNPs, methylation probes. Okay, so this is the methylation level. This is basically asking, is this region methylated or not methylated? And methylation is a form of is an indication of repression in gene regulatory regions. Okay, and then you can ask, well, where were these probes? And you can sort of see these arrows pointing, these lines basically telling you where in the genome were these probes. Everybody with me here? And then at the bottom, you basically have genotype. You basically ask, is this individual carrying 0, 1, or 2 of the reference alleles or of the risk alleles or of the alternate alleles? Okay, so this is the genotype matrix, again, 0, 1, 2. Everybody with me? So what is, and then these are mapping to the genome in the same way. So then what a QTL analysis does is look for genetic variants whose vectors here correlate with methylation probe vectors nearby. Sounds good? So we're basically just doing a correlation analysis in a local way that basically tells us, does the genotype here correlate with the methylation pattern there? Sounds good? It's super simple. Of course, to a first approximation. The genotype matrix needs to be corrected for these principal components that we talked about last time. Basically, depending on whether you are from Chicago or from Norway, your genetic vector will be influenced with thousands of loci that correlate with the geographic location of where you're from, which in turn correlates with all kinds of global predispositions to health and disease, some of which are ethnicity based, some of which are socioeconomic, some of which are location based, and so on and so forth. The type of healthcare that you have where you live, and so on and so forth. Okay, so the genotype matrix itself needs to be corrected by removing these principal components. The methylation matrix itself has also to be corrected by removing global effects. What are some of these effects? Well, you can basically ask, what is the effect of whether an individual has Alzheimer's or not? Or how long was the interval between when they died and when their brain was profiled? The post-mortem interval, or PMI. Which study did they come from? How many years of education did they have? What is the cell type mixture proportion? What plate was used in profiling these? What is the overall bisulfite conversion rate, which is basically telling you the experiment itself that determines methylation converts non-methylated Cs into something else, and therefore you're able to sort of measure methylation from that conversion. The longer you apply that conversion, the more of a global effect you have on methylation, or what was their age, what was their sex, okay? So for each of those variables, we can basically ask how much of the observed methylation variance across the whole matrix did that variable globally explain. Does that make sense? So I can basically ask for each of these known potential confounders, how much did it capture? And you can see here that the plate, the particular experiment where this sample was profiled, plays a very big role. The mixture, the inferred mixture of cell types in that brain sample plays a big role. The overall bisulfite conversion rate and the sex play a big role. But then the other confounders play a relatively smaller role, okay? Potential confounders. You can do that using a supervised approach by basically saying, what are the variables I already know about? Or you can do that using an unsupervised approach by basically taking your principal components or your singular vectors and then asking, if I take that whole matrix and I decompose it, how much variance does each of these vectors capture? Raise your hands if you're with me on this one. Great. And then what you can see is that different vectors capture different amounts of the total variance. And then as you start adding these vectors together, you get to more variance explained with the first 7 principal components than with all of the known or measured covariates. So that shows the importance of doing these global analyses that are completely unbiased, because these can capture many more confounders than the known covariates. Everybody with me? And then you can ask, well, how much additional did the known confounders capture that was not captured de novo? And you can see here that there's another 5% of the total variance that is captured there. And you can also do the same. You can basically say from all the stuff that the known covariates captured, how much more was there to be captured by the de novo covariates. Everybody with me? Great. So the first thing you do in your analysis is ask, how do I correct for all these global effects? And one way to do that is to basically say, how much of the variance have I captured in different regions of the genome using different types of normalization. And here you can basically see that as you're correcting for more and more of these principal components, you can basically capture more and more of the variance. And eventually there's this very sharp shoulder where the amount of variance captured decreases dramatically, and then that's probably a good place to stop removing these global components, because at some point you're going to start removing signal as well. Everybody with me? Yeah. Some variation of that. Yeah, they're not ordered by, you know, they're not PC1, PC2, but they're learned completely de novo. They're not based on that.
So if you're completely separate, how do you know that maybe like one of them doesn't correspond to you?
That's a great question. In fact, that's exactly what this matrix here tells us. This part of the matrix basically tells us how does a singular value vector 1 capture each of these known biologically measured variables. And what you can see is that you can partition your you can partition your singular vectors into the ones that are associated with plate. For example, 1, 3, 5, and 7, or 5 and 6 are associated with plate. Component 4 seems to be capturing something that has to do with cell mixture proportion, and component 2 is capturing something about the individual's age and sex combined. Does that answer your question? So basically that tells you the correspondence between this part and that part. Everybody with me? Awesome. I'm showing you this because as you carry out your own analysis, these are the types of things that you want to be aware of and able to do. The next thing we did is basically ask, how are these probes varying across individuals, and how is that variation related with the chromatin state annotation of the different regions? Most analyses are basically treating all methylation probes as equal. But what we found is that, in fact, the specific chromatin state that these probes fall in dictates a lot of their behavior. And in particular, if you look at the average methylation of different probes and the average variation in the methylation, like, for example, the standard deviation of methylation, we found that it varied dramatically between genetic variants falling in different types of chromatin states. In particular, you basically could see this bimodal distribution in the average methylation level of every probe. And that basically tells you that some probes are very highly methylated, some probes are very unmethylated, and very few probes fall in the middle. Okay? That, of course, has a very big impact on whether this probe varies a lot or a little. So you can basically see how much, sorry, a lot or a little. So you can basically ask, what is the overall standard deviation in the methylation of that probe across individuals? And what you see is that some probes vary by a lot and others vary by very little. And the ones that vary by a lot are the ones that also have more of an average mean rather than an extreme mean. And this kind of makes sense. Basically, a probe that is already quite high doesn't, or maybe I should put it differently, probes that don't vary, probes that have a very high average are very unlikely to be varying a lot, okay? And conversely, you have more room to change if you're here, whereas if you're unmethylated or fully methylated, there's much less room for change, okay? And then we also checked that this intermediate methylation was not simply an artifact of averaging bimodal events between individuals. You could basically say, well, hey, maybe this is average methylation because in half the people it's here and the other half of the people is here. Everybody with me on that? And basically what we found is that, in fact, no, it didn't appear to be that as you sort of looked at that distribution, if that was the case, you would find that the intermediate probes are in fact populating the entire spectrum here, but no, the intermediate probes are still mostly intermediate across all individuals, and they just vary by a little bit around that. Everybody with me? Great. The other thing that was actually quite striking is that if you looked at the probes that had the most amount of methylation and the most lack of methylation, you basically found that they largely corresponded to transcribed and promoter states. That basically the chromatin state annotation of these probes told us a huge amount about their average methylation. So transcribed probes were very methylated, and promoter probes were very unmethylated. And then in the middle, you basically had a huge overrepresentation of enhancer chromatin states. So basically probes that sit in enhancer regions were by far the most overrepresented in this intermediate methylation level. Everybody with me? So that basically says that if I know the chromatin state that you're in, I can say a lot about your, average methylation. But what's really cool is that this intermediate methylation state was also the one that showed the most variability. So you can basically see that if you ask how variable are different probes, you basically found that enhancer probes are by far the most variable, promoter probes are by far the least variable, and then there's a whole range in between. And if you look at the overall probe methylation variance, you basically find that there's an order of magnitude difference in the overall variance of the enhancer probes versus the promoter probes. Enhancers are 10 times more variable versus have a 10 times higher variance than promoters. Everybody with me? So remember when, in the epigenomics lecture, I was saying, well, promoters are kind of boring. They're kind of always on, whereas enhancers are extremely dynamic. Between tissues, they vary dramatically. Turns out that between individuals, it's kind of the same. Promoters are kind of boring. They're kind of always on, whereas enhancers vary dramatically between individuals, okay? So now we can basically turn to discovering quantitative trait loci. And this is the most basic relationship that you're looking for. You're basically looking for a place where knowing the genotype of a nearby genetic variant tells me a lot about the methylation level of that nearby methylation probe, okay? So if I have 0 alleles, 1 allele or 2 alleles of that, particular SNP here, RS2291199, then my average methylation changes dramatically, okay? This is quite a striking effect. Basically says that if I know the genotype of that individual at birth, I can predict the methylation level of that individual's brain at 93 years of age. This is extremely important for causality. Basically says that when that person is born, I could have predicted their methylation level. And therefore, if I can correlate that methylation level with disease, I have a pretty good chance of getting at causality, because I can basically say that the genetic variants play a very big role in determining that overall methylation level, which then correlates with disease and therefore, is likely to play a causal role in disease. Everybody with me? Again, that locality of QTL analysis is extremely important, because it doesn't just say, oh, from all the places in the genome. It says, no, from the ones nearby. And therefore, if there's no other probe that correlates with that globally, it tells you that the impact is most likely a direct one acting in cis, okay? So that was an example here. Here's an example down there, and down there. You can still see here that there's a p-value of 10 to the minus, even, all the way down here. And up here, the p-value is 10 to the minus 300. So this is a hugely significant association between genetic variation and intermediate molecular phenotypes. Yeah.
Just wondering about the traits and technical stands, right? So they're that line. Like, why are the ones above it?
Oh, all of these guys look even better than that.
Right.
Yeah. So basically, the only reason why I'm showing you an example down the middle is to be nice. Basically, I could have shown you 3 examples from all the way up there, and they all look even more dramatic than that. Does that make sense? So it's not that, oh, we discovered these guys. Like, no, all of these guys are amazing, including this one all the way here at the bottom. Okay. Yes. Thanks for your question. Any other questions? Yeah. We haven't yet looked at Alzheimer's. Right now, we're just looking at methylation. Yeah. So basically, right now, these are just methylation QTLs. The question now is how many of those methylation levels are in fact correlated with Alzheimer's disease. And then we can get at the contributors to Alzheimer's. But to answer your question a little provocatively, yes, sure. There's like probably 1,000 contributors to Alzheimer's disease, each of which has a tiny little effect. So the question is, how do we use all that information? Well, we can use it to figure out what are the pathways that are affected and how are these pathways converging into a small number of molecular biological processes, which we can then intervene into. The reason why we're doing all this exercise is not to basically say, oh, quick, there's a methylation change here. Let's go and intervene and change that methylation probe. No, because all of these are likely very weak effects. The reason we're doing all this is to basically identify the pathways that underlie the disease, the cell types where they act, and sort of then learn how to intervene on those pathways. Any other questions? Yeah.
The bottom plots, why are the clusters not exactly outlined?
That's such a great question. So you would have expected to see 0 with a bunch of dots, 1 with a bunch of dots, 2 with a bunch of dots. I already gave you the answer. Can anybody sort of figure out from what I said earlier as to why they're not 0, 1, and 2? What did we do on the genotype matrix? We corrected for these principal components. When you correct for these principal components, that basically means that your genotype matrix itself is no longer a bunch of 0s, 1s, and 2s. They're now shifted according to that correction. Does that answer your question? So that, you know, for every single one of these probes and SNPs, this is correcting for these global principal components. Yeah. Correct. Aha. So, like this. So, how nearby is your question? Well, nearby could be, you know, 20 nucleotides. It could be 1,000,000 nucleotides. So, which is it? The answer is, as you go further and further, fewer and fewer of these probes fall at that distance. So I can basically say at exactly on the probe, what fraction of my SNPs that are QTLs for that probe are located, and at 50 kb, what fraction are located there, and so on and so forth. What you can see here is that 90% of your methylation QTLs fall within 75 kb, 75,000 nucleotides, okay? So the genome is a big place. 3 billion bases is a lot. So even 1 megabase is 0.3% of the human genome. So that's, you know, 300 kb, that's 0.1% of the human genome. So they're extremely, extremely tightly packed. And then you can basically see that whole distribution and how far away the probes are in general. And you can see here that even for, so when I said 75 kb, yeah, that's exact. So it's a radius of 150 kilobases. So that's 0. So it's half of 1 per 1,000 of the human genome. Okay, if I can do math in my head, which might not be the case in lecture. All right, so then you can basically ask, how does my discovery power increase with the number of individuals? If I have 25 individuals, 50, 100, 150, 200, 250, 300, 400, all 600 that had both genetic variation and methylation variation, how many QTLs have I discovered? And here you can see that with 25 individuals, I discover 138 QTLs, and they're mostly nearby. With 50 individuals, I discover 2,500 QTLs, and they're still mostly nearby, and they mostly have very strong effects. And then as I increase the number of individuals, you can see that I'm discovering weaker and weaker effects. Basically, I have the ability to detect significantly at a significant threshold, you know, weaker and weaker effects. So basically, much more subtle effects I'm able to discover, and also I'm able to find things that are further and further away. Everybody with me with these plots? And then you can ask, what is the number of methylation QTLs that I've discovered as I go to more and more individuals? So here, the number of individuals allows me to discover many, many more QTLs and have the minimum detection threshold is lower and lower, so it can discover more and more subtle effects. And then the green one basically tells you what is the median effect. And the green one basically tells you that, yes, I find many more, but their effects are more and more subtle. Sounds good? The other question you can ask is, how reproducible are these effects? Namely, yay, I'm so excited, I found some eQTLs with 25 individuals. If I now go to 500 individuals, are these still significant? So what is the validation rate of these methylation QTLs? How many turn out to be real? And the answer is quite abysmal. Namely, you can ask, how much did I misestimate the effect of that QTL? And if my effect that I predicted was, I don't know, 0.85 or something like that, then my average misestimation is about 0.5. So basically, that's as bad as it gets, and the confidence intervals on that are just, again, abysmal. So that basically means that I can't really believe any of these methylation QTLs that I discovered with very, very few individuals. But as I go to 50 and 100 and 200 individuals, then the average misestimation is dropping dramatically again. And you can see here that with 100 individuals, I'm on average off by 0.1, which is not so bad. Everybody with me? Who feels that they're learning stuff? Yeah, good. Okay. The other questions, of course, what is the ascertainment bias in the allele frequencies? Because with few individuals, I'm only able to look at very high allele frequencies. If I only have 25 individuals, I'm never going to discover a methyl QTL whose minor allele frequency is less than 1 in 25, right? Because that means I will only have one person there. So if you look at how the minor allele frequency of the discovered methylation QTLs changes as I increase the number of individuals, you can basically see here that it starts out extremely skewed for very common variants, and then that sort of shifts down progressively to being quite flat. And then you can ask, well, what is the background distribution of minor allele frequencies? And you can see here that it's in fact exactly the reverse. The more infrequent you are, the more common it is that you are this infrequent. Okay. So basically, most SNPs have very low frequencies, and then this is cut off because in reality, it sort of boosts up dramatically. But here it's kind of flat. Yes, we're still preferring to discover things that are higher frequencies, just because that's where we have most power. Everybody with me? Any questions? Okay. So then, of course, you can do the same question with methylation QTLs. You can basically say, yeah, great. Promoters were kind of boring when it came to tissue variation, and enhancers were very, very exciting. Promoters were kind of boring for inter-individual variation. Enhancers were very exciting. Now you can ask, well, what about genetically driven variation? And again, you see that promoters are kind of boring. They're kind of depleted for methylation QTLs, whereas enhancers are very exciting. They're very enriched for methylation QTLs. So they're much more likely to be affected by genetic variation. And part of the reason for that is that if you look at a promoter, so many different processes converge on that promoter and basically keep it stable. One SNP is very unlikely to have a very big effect. Whereas in an enhancer, it's kind of like out there. And one SNP could actually have a big effect. Is everybody with me, at least about the intuition about what could be going on here? So it's not that promoters are not important. Promoters are important, and there's a lot of regulation that happens there, but they're much less likely to be disrupted by a single SNP. They're much less likely to vary across individuals. They're much less likely to vary across tissues. If you were to ask me, where do I put a bomb? Do I put it in an enhancer or in a promoter? I'd rather put in a promoter. That's much more likely to have a big effect. But SNPs are not bombs. They're like tiny little, fluctuations. And these tiny little fluctuations are much less likely to have an impact on disease in promoter regions. There's, of course, another argument, which is evolutionary, which is that evolution selects against big bumps. So basically, if a big bump happens in a promoter region, i.e. a genetic variant that will dramatically alter that promoter region so that that gene is no longer ever expressed, well, evolution is probably not going to leave that variant around at high frequency. Same thing with the protein coding regions. I mean, bumps, i.e. strong effect SNPs, are very much excluded from protein coding regions. Okay? Everybody with me here? Great. And then we can ask how informative, given that promoters are depleted and enhancers are enriched, if I were to focus on different chromatin states, what would be the total predictability that I would have on disease? And the answer is that if I take genetics alone, all of the SNPs in the genome give me about 59% area under the curve for predicting Alzheimer's. If I add to that all of my covariates that I mentioned earlier about education, age, sex, etc., then I get to 69%. And then the question is, compared to that baseline of 69% with all of the covariates, how much is methylation alone giving me? And the answer is, there's another 4% of the variance that I can explain using methylation. And that's taking all methylation in the genome. If I instead focus on only promoters, I explain much less. And if I instead focus on only enhancers, I explain much more. Okay? And the reason for that is that there's a lot of noise that's added with promoters. Whereas if I take away all these noisy predictors, I'm able to get higher predictive power by focusing in only on the probes that are the most informative. And therefore, my predictor does not have as much noise. Everybody with me? And then you can basically ask, if I rank all of my probes according to their association with Alzheimer's, how enriched are enhancers versus promoters? You can see here that enhancers are much more enriched, and so on and so forth. Okay? So that basically allows us to now start asking, if I have the ability to predict methylation from the genome, can I start correlating that predicted methylation with the disease phenotype? And the answer is that this helps you dramatically discover many new loci that are associated with disease. So again, just to sort of clarify the differences here, so a genome-wide association study is basically going from genetics all the way to disease directly. A methylation QTL study is basically asking how is genetics affecting this intermediate molecular phenotype. An MWAS, or methylome-wide association study, is basically asking about the correlation between intermediate methylation levels and disease. But that's a bidirectional arrow, as we mentioned earlier, because this is correlation, not causation. Some of these methylation changes might be downstream of disease, or they might both be downstream of some environmental change. Whereas if I instead use the genetics to predict methylation, and then use the predicted methylation, i.e. the genetic component of methylation, to correlate that with disease, that genetic component is a unidirectional arrow. Everybody with me? Because again, I only look for local genetic effects, much less likely to be coming from some transacting factor that is acting across all genes, and therefore I get my causality back. Sounds good? So applying this to Alzheimer's, we're able to find many more genetic loci that are having a predicted causal effect on Alzheimer's through their methylation impacts. Namely, you can basically look at GWAS, and the threshold that you need for the multiple hypothesis testing is above the top of this graph. So 10 to minus 2, 4, 6, 8 would be somewhere around here. Okay. So all of these are not genome-wide significant. That's across all of chromosome 6. And instead, if you take these genetic variants and you predict intermediate methylation levels, and then you do the correlation between methylation for this much smaller number of probes and disease, you basically find that there are, half a dozen genome-wide significant loci after bone-forming correction whose line is down here, the dotted line. Everybody with me here? So basically, intermediate phenotypes are extremely important for mechanistic insights on disease. Number one, they give you the intermediate phenotype. Number two, they tell you what tissue you might be acting in, what stage in life, what region, what pathway. And lastly, it also allows you to discover new loci that may not be genome-wide significant. Sounds good? Who feels that they've learned some so far? Awesome. So let's now step back and see what are the basics of doing such a QTL analysis, okay? So let's basically now go back to gene expression as a quantitative trait. We saw an example on methylation. We saw earlier an example on gene expression. Let's now go back to eQTLs, not methyl QTLs, but eQTLs. So the idea is we would like to be able to find cis-acting genetic variants where the particular number of alleles that you have of T, for example, has impact on the expression of a particular gene, okay? So this is, one such example, okay? So gene expression itself is a quantitative trait. So you can basically ask how heritable is that quantitative trait? And you can basically see that there's quite a lot of heritability for gene expression levels. So what are the methods that we use for eQTL discovery? The most basic one is the following. It basically says, can I explain my expression as a function of my genotype? So basically, can I have an output variable y, which is the quantitative trait of gene expression, as a function of a genetic variation of a nearby SNP? And then, of course, this is the coefficient with which this acts, and then this is the y offset with which this acts, and then some error surrounding it. Okay? So you're basically looking for a correlation between expression and genotype. And because the genotype is unlikely to be the one influenced, this gives you causality between genetics and expression. Sounds good? So basically measure expression across hundreds of individuals. I measure genotype for the same individuals. I do all kinds of corrections for both. And then I look at this very simple equation. So, one way to do normalization is to have some filler, some transcripts that are based on, spike-ins. And then for the covariates as well, for the genotyping, I'm correcting for all kinds of principal components. And then I'm also including in my linear regression additional explanators for how the expression could be changing. Namely, if expression is simply changing due to age, gender, or population stratification, or technical covariates, I want to remove that first. So I can remove all these components from my expression matrix. Okay? There's an important consideration to have, which is if the probe itself, if your methylation probe or your gene expression probe is an array, and that SNP completely changes the hybridization between that probe and your sequence, that's not a change in expression. That's simply a change in hybridization. So I can't even measure transcripts that don't have an A here. So you should remove any kind of SNPs that directly disrupt your probes. Is everybody with me here? In the same way, genome alignment is lower with mutations. If I have a reference allele, I'm much more likely to align to that than if I have an alternate allele. I'm much less likely to align to that. So very often I have to align directly to an alternate genome, which is personalized for that individual, or I have to discount for differences in alignment rates when a SNP is there. Is everybody with me on that? Lastly, if a gene itself is extremely variable to start with, then changing that mean by a little amount doesn't tell me much. But if a gene has a very tight expression, then changing that mean expression by a little amount tells me a lot. Everybody with me? So basically, the overall variance in the expression or in the methylation of a gene has a dramatic effect on my ability to discover QTLs. Another one is basically how far do I look? Do I only look at, I don't know, 50 kb or 100 kb or a 1 megabase window nearby? And do I only look at cis? Do I look at all SNPs and trans? And how do I correct for demographics, population stratification, technical covariates, and so on and so forth? So adjusting for all of these technical covariates, in fact, dramatically increases the ability to discover these eQTLs. If I correct for no covariates, then I discover 2,500 QTLs. If I correct for about 10 covariates, then I discover about 3,500 QTLs. So dramatic increase in the number of QTLs. That basically means that there was a lot of non-genetically driven variation that I'm correcting for. Of course, as you start including more and more principal components, you're chopping away at the signal in the array. Eventually, the number of QTLs decreases dramatically. Everybody with me here on sort of this shape of the curve? So basically, initially it's increasing because I'm removing non-genetic components, and then it's decreasing dramatically because I'm actually starting to remove genetic components as well. Sounds good? So what I'm effectively doing is not just alpha plus some, slope times the genotype, but I'm also doing some slope times gender, times age, times all the principal components of the genotype, plus all the principal components of expression. And ultimately, I'm basically asking what is my, how likely is this model to explain the effect versus the same exact model, but without genotype. Okay, so that's exactly what a QTL test is. If this is a true QTL, then this component here in the context of all the rest is giving me additional information. Sounds good? So we talked about the minor allele frequency. We talked about sort of different normalizations. We talked about how wide of a search do I make and sort of the effect that these can have. Okay, so the minor allele frequency can basically give you more or less QTLs that you're discovering. The further you go into rare variants, the more noise you have, but the more variants you're also discovering. And then setting the parameters is tough. Basically, knowing how many components to remove is hard. Okay, and then the specific combination of these parameters might be extremely data set specific. So for one data set, these might be the right parameters, and for another data set, it might be completely different parameters. Okay. So you can actually tune this QTL discovery to that. So there's a lot of insights that we can already gain about eQTL function and biology. So the first is that focusing on eQTLs has a huge advantage in being able to discover true disease-causing variants. The second one is that these eQTLs are positioned very, very close to the target gene that they're capturing, as we saw earlier. The third is about reproducibility. In different cohorts, the effect size might vary, but generally the direction of effect is the same. They are very often tissue-specific, but there's quite a bimodal distribution. Some QTL SNPs are very common across tissues, and others are extremely tissue-specific, but there's very few in the middle. So basically, the same QTL might simply be an extremely cell type specific or extremely tissue agnostic QTL. If you look at the genes downstream of that, it's very important to be able to sort of identify the downstream target genes. You can basically ask about the co-regulation of multiple genes controlled by the same locus. And this is actually very common, where the same SNP is in fact having a genetic effect on the expression of multiple nearby genes. And then this has to do a lot with co-regulation, these functional domains. Transacting QTLs are very difficult to discover. And the reason is that the hypothesis space is so much larger. You need to do a lot more correction. And you can use graphical ways of discovering them by basically saying that a QTL would likely be a QTL for many genes in the same pathway. through some transacting factor that it controls. They're very enriched in disease, and they allow you to sort of start predicting causal variants much more frequently. So if you condition on something being an eQTL, your odds ratio of truly being a GWAS SNP is dramatically increased, much more than many other priors. And then you can increase GWAS variant prediction by focusing specifically on QTLs. Instead of saying I'm going to discover any SNP in the genome agnostically, you can say I'm only going to focus on eQTLs, and then that leads to dramatically increased discovery power. The last thing that I want to touch upon is allele-specific analysis. So eQTLs are basically looking at the difference between the dosage of GG, GA, AA. How many copies of the minor allele do I have? Everybody with me on that, right? Now, what allele-specific analysis does is look within this middle group of individuals that are heterozygous. For those individuals, one chromosome inherited from mom has one allele, and the chromosome inherited from dad has the other allele. So the GA carriers are in fact the ones that have differences between their two chromosomes. This is very powerful because I can now use sequence identity of the SNP, of the reads that are coming from mom's chromosome and from dad's chromosome, to basically see if there's a difference in expression within every cell of that person. And the beauty and the power of that is that I'm controlling for all of the transacting variables. Because I'm basically saying, for this one individual where everything else is the same, do their two chromosomes show different expression? Okay? I'm not just simply saying, who are all the AA individuals and who are all the GG individuals and are they different? The problem with that analysis, with QTL analysis, is that they may be different because of disease. And that might be impacting the expression level. But if I ask within GA individuals that have both alleles, what is the difference between one copy and the other copy in gene expression level, then I'm controlling for everyone else, for everything else. Raise your hands if you're with me on this one. Awesome. So what allele-specific analysis does is look specifically within heterozygous individuals for differences in expression between one allele and the other allele. And this is, very, very powerful. And you can combine that with the QTL approach. You can basically say what is the total QTL effect and what is the allelic imbalance between them, and sort of have a much more powerful test that combines multiple haplotypes. Okay? So that's where we're going to stop today. So basically we talked about motivations for mapping regulatory variants. We saw an example of carrying out a whole QTL analysis in the context of Alzheimer's disease and all the things we can learn, and the impact of allele frequencies, and how to do the corrections for genotype, how to do the corrections for experimental principal components. And then we saw the diversity of methodologies for QTL discovery. And then we saw the, some insights that are inferred. And lastly, how we can carry out allele-specific analysis. We're going to talk about mediation and Bayesian inference in a later lecture. Who feels that they've learned stuff today? Yay, awesome. Great, see you guys on Thursday for systems genetics.